Okay, can somebody confirm you can hear me,
I
please?
can hear you.
Thank you. Before we begin, are there any questions? Today is May 20th. Let me share my screen here. And we are supposed to be working on chapter 8. I think we did get started. For the lab today, there will be a lab 9, our last real lab. Note that we have a quiz this Wednesday, quiz 4. Quiz 4 will be covering lab 7 and chapters 5 and 6. Any questions about anything we're doing? Let me remind you, you should be working on your unknown project. Not many people have gotten started on it, and I realize that you were probably working on your infectious disease project. But now that you've got that turned in, please work on your unknown project. Most of you will take about 2 weeks to finish that project. So you cannot get it done in just a few days. Any questions about any of that? No, not the lab. Let's go to the lecture. We didn't get very far. We only got onto chapter slide 6. All right, I talked about DNA and mentioned that it was a single strand molecule, and two of them run anti-parallel to make the double-stranded DNA. They're only held together by hydrogen bonds. At least each strand is only held together to the other strand by hydrogen bonds. When we look at the flow of information within a cell, it's said that the information flows vertically from DNA to RNA to protein. So in DNA, this is known as the central dogma. The information is stored in DNA. It's then transcribed into RNA and then translated into protein. In each of these molecules, the information is stored in a different language, and we'll talk a little bit about that. So that's within a cell. The information flows from DNA to messenger RNA to protein, but you can look at the information flowing between generations of cells, or for humans, we could say between parent and offspring. But it flows from a parent cell to the DNA, then replicates and divides into two and moves to opposite ends of the cell, and the cell divides into two daughter cells. Uh, for humans, it would be the sperm and the egg would unite, meaning you don't have the DNA replicating exactly. It does to make the sperms in the egg cell, but once they're made, the DNA doesn't replicate. Then the cells unite, and then they form one offspring, which will be the zygote, the first cell of the offspring. Any questions about any of that? You can also look at and that's the flow of information between generations of cells. You can also look at the flow of information between cells of the same generation. And I've broken the chart here because I didn't like the way it was going. The DNA flows out of one cell, and then you have naked DNA outside the cell. It then goes in a recombinant cell. And this DNA is DNA not from this cell, but from this cell. And that is known as recombination or the flow of information between cells of the same generation. Any question about any of that? All right, so the vertical flow of information within a cell starts in DNA, And the language that that information is stored in is called nucleotides, the DNA nucleotides. And there are only four different nucleotides. So there's only four letters in the alphabet. For RNA, the information flows to RNA, and that information is stored in the RNA nucleotides. Slightly different than the DNA nucleotides, but once again, there's only four letters in the alphabet, meaning there's only four different RNA nucleotides. And then the information is stored in RNA is flows to protein, and that information is stored in the amino acids. And there are 20 different amino acids, or 20 different letters in the alphabet of amino acids. For comparison, English has 26 letters. Any question about any of that? So if the information in the DNA is that is a dog, that's the original information in the DNA. In RNA, it would be similar, but it would be a different dialect. That is a dog. And then in the protein, the amino acids, the information would be something like quello è un cane. Totally different language. Any question about any of that? All right, that's enough for the flow of information within a cell. Let's talk about the flow of information between generations of cells. This is also known as the vertical flow of DNA. The horizontal flow is this to the recombinant cell, or between generations of the same between cells of the same generation. In the flow of information between generations of cell, the DNA from the parent cell replicates. Each DNA molecule moves to one end of the cell, and then the cell splits down the middle to make two daughter cells. Both of them are copies of the parent cell. Any question about any of that? How that happens is the DNA molecule, which is double-stranded, becomes single-stranded, and then one strand is used as a template to make the other new strand, which is the same as the strand over here. And let me blow this up so I can show it a little better. So when the DNA is a single strand, the DNA polymerase will bind to this nucleotide and see that it's a T here, and it'll put in the corresponding nucleotide, which is always an A. And then the DNA polymerase will move down to the next nucleotide, see a G there, and add a C on this growing strand of DNA. And then the nucleotide will move down to the next nucleotide, which is sees as a C, and it'll put the corresponding G on the new strand. So one strand is used as a template to make the new strand of DNA. Any questions about any of that? This means the DNA replication is semi-conservative, where one strand is used as a template to make the new strand of DNA. That differs from the conservative model, which would have here are the dark strands of DNA or the old strands of DNA. The white strand is the newly replicated strand. And in the semi-conservative model, each new strand, double strand of DNA will have one original strand and one newly replicated strand for both cells. The conservative model would be that one cell would have the old parental dark strands, and then the new cell or the dividing cell would have the both white strands of DNA. That's the conservative model. The conservative model doesn't happen, only the semi-conservative model happens. And that is one strand is used as a template to make the newly replicated strand. And don't ask me what the liberal model is because I don't know. Any questions? No, I need to verify my account again. I think we already did this at the start of the term. So when the DNA is building a new strand of DNA using the template, the DNA polymerase will bind to this nucleotide, see it's an A, and then add the corresponding T. How that happens is a T triphosphate gets added, so all these bases that are added will be a nucleoside triphosphate, and then the bond will break right here. That's a high energy bond, and that allows this T to be added to this growing strand of DNA, meaning that adding that takes energy, and where the energy comes from is actually not from ATP. It is from a nucleotide triphosphate, like right there would be ATP adding, but it doesn't break the last strand of the phosphate. It actually breaks the second strand of the phosphate. And then the phosphate binds to the ribose here. And then this makes a covalent chemical bond holding the single strand of DNA together. Any questions about any of that? And that shows right there how the reaction occurs. The dipyrophosphate comes out, meaning the two phosphates, and the T is added here, and the phosphate here binds to the ribose there. Any questions about any of that? All right, let's look at a little video of DNA replication. To get the DNA replicated, remember you have to make the DNA single-stranded.
DNA is a molecule made up of two strands, twisted around each other in a double helix shape. Each strand is made up of a sequence of 4 chemical bases, represented by the letters A, C, G, and T. The two strands are complementary. This means that wherever there's a T in one strand, there will be an A in the opposite strand, and wherever there's a C, there will be a G in the other strand. Each strand has a 5' end and a 3' end. The two strands run in opposite directions. This determines how each strand of DNA is replicated. The first step in DNA replication is to separate the two strands. This unzipping is done by an enzyme called helicase and results in the formation of a replication fork. The separated strands each provide a template for creating a new strand of DNA. An enzyme called primase starts the process. This enzyme makes a small piece of RNA called a primer. This marks the starting point for the construction of the new strand of DNA. An enzyme called DNA polymerase binds to the primer and will make the new strand of DNA. DNA polymerase can only add DNA bases in one direction, from the 5' end to the 3' end. One of the new strands of DNA, the leading strand, is made continuously, the DNA polymerase adding bases one by one in the 5' to 3' direction. The other strand, the lagging strand, cannot be made in this continuous way because it runs in the opposite direction. The DNA polymerase can therefore only make this strand in a series of small chunks called Okazaki fragments. Each fragment is started with an RNA primer. DNA polymerase then adds a short row of DNA bases in the 5' to 3' direction. The next primer is then added further down the lagging strand. Another Okazaki fragment is then made, and the process is repeated again. Once the new DNA has been made, the enzyme exonuclease removes all the RNA primers from both strands of DNA. Another DNA polymerase enzyme then fills in the gaps that are left behind with DNA. Finally, the enzyme DNA ligase seals up the fragments of DNA in both strands to form a continuous double strand. DNA replication is described as semi-conservative because each DNA molecule is made up of one old, conserved strand of DNA and one new one.
Okay, any questions about that video? We'll talk more about it in the next couple of slides, but the three important enzymes to follow in DNA replication are helicase. It's the enzyme that opens up the two strands of DNA and makes it a single strand. The second enzyme that's important is the DNA polymerase. It's what makes the new strand of DNA, adding the nucleotides to the newly growing strand. And then the third enzyme, which is really important, is ligase. It connects the single strand of DNA together, meaning when the DNA polymerase, let's say the DNA polymerase were to fall off right here, and then another DNA polymerase were to bind right here, continuing the process. The ligase would ligate that old new strand of DNA to the new strand of DNA, meaning where the DNA ligase begins, not the DNA ligase, where the DNA polymerase begins, it would not link up the strand of DNA to the previous strand. For the newly replicated, what do you call it, leading strand, if the DNA polymerase never falls off and begins at the end of the chromosome, meaning the end of the strand of DNA, never falls off and makes the entire strand of DNA all the way, DNA ligase may not ever work on the leading strand. It would only work when the DNA polymerase falls off and then another DNA polymerase combines or binds to the single strand DNA and then continues the process. Ligase would need to link that strand of DNA to this strand of DNA. Is that clear to everyone? So on the leading strand, ligase may not ever work. If one DNA polymerase synthesizes the entire leading strand, DNA ligase doesn't need to come in. It's not needed. However, on the lagging strand, each Okazaki fragment has to be linked to the previous Okazaki fragment. Let me see, which way are we going here? So we're going this way. So this Okazaki fragment has to be linked to that Okazaki fragment. This Okazaki fragment has to be linked to that Okazaki fragment. And so ligase is working quite a bit on the lagging strand of DNA. Any questions about any of that? Now, the reason why the lagging strand has these Okazaki fragments is because it's running in the wrong orientation, and the DNA polymerase wants to run in the 5' to 3' direction, and this one isn't running the 5' to 3' direction. It's running 3 to 5', so not 5 to 3'. And so the DNA is synthesized not this way, the way the helicase is moving. The DNA is made the opposite direction, and so it's made in short chunks. and they were discovered by Dr. Okazaki, and that's why they're called Okazaki fragments. Any question about any of that? Let's see. So I talked about the leading strand of DNA. I talked about the lagging strand of DNA. I talked about the helicase, opens up the two strands of DNA. DNA polymerase makes the newly replicated strand using the template as the old strand of DNA is the template. And then the third important molecule is the ligase, which will link the broken pieces on the newly replicated DNA together, which on the Okazaki fragment, ligase has to work on both ends of the Okazaki fragment, linking it to the Okazaki fragment nearest to it. And then the ligase will work on the leading strand because frequently the DNA polymerase falls off. And then another DNA polymerase will bind to the DNA and continue the process. And where that newly replicated strand began, it will not be connected to the other replicated strand of DNA. So ligase has to link it together. Any questions about any of that? All right, I did mention that DNA is made in the 5 to 3 prime direction. It is initiated by an RNA primer. That's not as important because the RNA primer will be removed. The leading strand is synthesized continuously because it runs 5 to 3 prime. The lagging strand is made discontinuously because it runs not in the 5 to 3 prime direction, and it results in the Okazaki fragments. Then the RNA primers are removed, and DNA polymerase fills in the RNA primer, meaning that's actually RNA in the RNA primer. It's not DNA. And you need a DNA polymerase to put in the DNA. And then DNA ligase finishes the work by linking the newly replicated strands of DNA on the single strand together to make the single strand of DNA continuously linked by covalent chemical bonds. Any question about any of that? All right, so that's DNA replication. Let's move on to RNA transcription. The synthesis of RNA from DNA is called transcription. DNA is transcribed to make messenger RNA, tRNA, or rRNA. How does the let me see if I can remember this right. How does the RNA polymerase, when it's reading the DNA, know to make messenger RNA, tRNA, or rRNA? That's a very simple question. And if you don't know it, you should learn your genetics a little better. How does the RNA polymerase, when it's reading DNA, know to make messenger RNA, tRNA, or rRNA? I should probably ask this as a quiz question because students don't quite seem to understand. It's a very easy concept, too. The RNA polymerase does not need to know to make messenger RNA, tRNA, or rRNA because the RNA polymerase is reading the DNA, and if the DNA codes for a protein, it will make messenger RNA. But if the DNA codes for tRNA, the RNA polymerase will make tRNA. And if the gene codes for rRNA, then the RNA polymerase will make rRNA. Any questions about any of that? So the answer is it depends on which gene the RNA polymerase is reading. If it's reading a tRNA gene, then it'll make tRNA. Any questions about any of that? And that brings in another point here, and that is unlike DNA replication, let's go back to the picture, where all of the DNA is replicated, in transcription, not all of the DNA is replicated. Can anyone guess which DNA is replicated and which is not? I'll give you an extra credit point because I've already told you the answer. Anyone have the extra credit point? All right. Only the genes are transcribed. The non-coding region of the DNA, meaning the DNA which does not code for a gene, is not transcribed. Only the genes are transcribed. In eukaryotes, most of the DNA is non-coding DNA, meaning most of the DNA does not code for a gene. In prokaryotes, it's the reverse, or most of the DNA codes for genes. There's very little, at least in bacteria, there's very little DNA which is not a gene. Transcription begins transcribing the DNA when the RNA polymerase binds to the promoter. A promoter is a region upstream of the gene that codes for the RNA polymerase to bind here. So the RNA polymerase binds to the promoter, a region of the DNA upstream of the gene. And then that RNA polymerase moves down the gene, or moves down the DNA, transcribing the DNA into RNA. And it follows the 5 to 3' direction, meaning transcription occurs 5 to 3' direction. RNA is also made 5' to 3'. And then the RNA polymerase continues transcribing the gene until it reaches the terminator. The terminator is a region at the end of the gene that codes for the RNA polymerase to fall off the DNA. So all genes end at a terminator, and that terminator codes for the RNA polymerase to fall off the DNA, and that ends transcription. All right, any questions about that? Here we're actually looking at an microscopic image. It's sort of an electron microscopic image, but it's actually a fluorescent microscopic image, not a regular electron microscopic image. Looking at RNA polymerase, that is the DNA. That little strand there is the RNA, which is beginning to be made off of the DNA that we call transcription. Any questions about transcription? All right, let's go through transcription in more detail. As shown here, we'll blow it up. Let's let your little video on transcription. Looks like we're going to get a commercial.
You know Harbor Freight for affordability, but durability goes right along with it. You see, we test and
proteins are contained in our DNA. DNA contains genes. A gene is a continuous string of nucleotides containing a region that codes for an RNA molecule. This region begins with a promoter and ends in a terminator. Genes also contain regulatory sequences that can be found near the promoter or at a more distant location. Motor region of the gene functions as a recognition site for RNA polymerase to bind. This is where the majority of gene expression is controlled, by either permitting or blocking access to this site by the RNA polymerase. Binding causes the DNA double helix to unwind and open. Then during elongation, the RNA polymerase slides along the template DNA strand. As the complementary bases pair up, the RNA polymerase links nucleotides to the 3' end of the growing RNA molecule.
Let me stop right here and then point something out. And that is initially when this RNA is being made, initially that RNA is binding to the DNA. It's a hydrogen bond. But after a while, the RNA falls off and it's no longer hydrogen bonding to the DNA. That allows the DNA actually to snap back together down here, meaning this DNA will bind hydrogen bond to that strand of DNA. Any questions about any of that? So let's back up a little bit.
End of the growing RNA molecule. Once the RNA polymerase reaches the terminator portion of the gene, the messenger RNA transcript is complete, and the RNA polymerase, the DNA strand, and the messenger RNA transcript dissociate from each other. The strand of messenger RNA that is made during transcription includes regions called exons that code for a protein, and non-coding sections called introns. In order for
Okay, for that exons and introns, these are only found in eukaryote cells. We'll talk about that near the end of transcription.
the messenger RNA to be used in translation, the non-coding introns need to be removed, and modifications such as a 5' cap and a 3' poly A tail are added. This process is called intron splicing and is performed by a complex made up of proteins and RNA called a spliceosome. This complex RNA strand that can leave the nucleus through a nuclear pore and enter the cytoplasm to begin translation. How is the information in the mature messenger RNA strand translated into a protein? The nitrogenous bases are grouped into 3-letter codes.
Let's stop there too. All right, any questions about any of that? So once again, the double strand of DNA has to be made single strand DNA for transcription to occur. It's very similar to DNA replication. And how that gets made into single strand of DNA is helicase opens up the DNA. So helicase opens up the DNA both for transcription and for DNA replication. So that's another enzyme which is important. And then the RNA polymerase is the other enzyme that's important for transcription. That's a little different than DNA replication, where we have a DNA polymerase that's involved in DNA replication. So transcription begins when the RNA polymerase binds to the promoter region of DNA. I don't see that on here. Oh, right there it is. So RNA polymerase will bind to the promoter. The helicase will then open up the double-strand DNA, and it's trying to show you this. Let me blow this up a little. It's trying to show you that initially when the RNA is transcribed, it will bind double by hydrogen bonds to the DNA for a little while. But after a while, that RNA falls off the DNA, and that allows, as this moves down this way, by the RNA falling off, it allows the DNA to snap back together with the second strand of DNA. And then the RNA polymerase moves down the gene, transcribing it, until the RNA polymerase reaches the terminator, which is at the end of the gene. The terminator codes for the RNA polymerase to fall off the DNA, and that will release the RNA molecule from the DNA and the RNA polymerase. The RNA polymerase is then free to bind to another promoter and continue the process. And of course, when the RNA polymerase falls off, the single strand of DNA will snap back together to form double-strand DNA, which in reality, I don't know if you can see where my hand is, so I'll use the mouse. If the gene started here, actually it started more like here, this region of the gene is already snapped back together, meaning it's double-stranded. It's only this portion here which is single-stranded. But it will become double-stranded once the RNA falls off and the RNA polymerase falls off. Any questions about any of that? All right, if not, let's talk about translation. Translation is the synthesis of proteins at the ribosome using the information in the messenger RNA. Messenger RNA is translated in codons. A codon is a group of 3 nucleotides. 3 nucleotides code for one amino acid, so a codon codes for an amino acid. Any question about any of that? Surprisingly, protein translation does not begin at the start of the messenger RNA. The ribosome will bind to the messenger RNA and will run down it, kind of reading it, but it will not begin protein translation until the messenger RNA codes for the start codon, which is always AUG, coding for the start codon. And AUG does a second function, and that is it also codes for adding the amino acid methionine. That means all proteins always start with methionine. And if the protein doesn't want the methionine on the protein as the first amino acid, then that first amino acid has to be removed. Any question about any of that? So all proteins, when they first get made, they always start with methionine because that's what the start codon codes for. And there's probably many proteins that don't want the protein to start with methionine, so it's just removed. And then the ribosome will move down the messenger RNA, translating it until the ribosome reaches one of the 3 stop codons. You don't need to memorize these because there's 3 of them. You should memorize the start codon because there's only one, AUG. But there's 3 stop codons, like UAA is one. And when the ribosome reaches the stop codon, it will stop protein translation. The ribosome will fall apart. The messenger RNA will be released. Any questions about any of that? All right, let's talk a little bit more about the codons. The genetic information in the messenger RNA is read in groups of 3. We call it a codon. So each codon codes for one amino acid. But organisms typically have about 20 different amino acids. Humans have exactly 20 amino acids. Some bacteria do have a different number, but most have about 20. And usually if they have a different amino acid, they just throw out, meaning they have a different amino acid that humans don't have, they throw out one to replace the one that's put in. So most living organisms have about 20 different amino acids, but there are 64 possible codons. Why? Well, there's more than one codon for an amino acid. For example, phenylalanine is encoded by both the codon UUU and UUC. Serine is encoded by 4 different codons, meaning UCU, that codon UCC, UCA, UCG, all of these 4 codons code for the same amino acid, serine. There we go. I had trouble getting my mouse to work. And then leucine, there's actually 6 codons that code for that one amino acid. there's only two amino acids which are encoded by one codon. The first is methionine. It's only coded by AUG. And the second is tryptophan. You don't need to know tryptophan. UGG is code for tryptophan. You don't need to know tryptophan either. You probably should know methionine, but tryptophan you don't need to know. And you don't need to know phenylalanine has only 2 codons, leucine has 6, serine has 4. You don't need to know that. I'm just pointing out to you that each codon or each amino acid may have more than one codon. Most of them have more than one codon. There's only 2 that have 1 codon. Some of them have 2, some of them have 4, some of them have 6 codon, coding for that one amino acid. This is known as the redundancy or the degeneration of the genetic code, where there's more than one codon coding for the same amino acid. Degeneracy of the code. That means degenerate more than one for the amino acid. Any questions about any of that? Let's watch a little video on translation.
Translation is the synthesis of a protein from an mRNA template. This process involves several key molecules, including mRNA, the small and large subunits of the ribosome, tRNA, and finally, the release factor. The process is broken into 3 stages: initiation, elongation, and termination. Let's see the process in action. Eukaryotic mRNA, the substrate for translation, has a unique 3' end called the poly-A tail. mRNA also contains codons that will encode for specific amino acids. A methylated cap is found at the 5' end. Translation initiation begins when the small subunit of the ribosome attaches to the cap and moves to the translation initiation site. tRNA is another key molecule. It contains an anticodon that is complementary to the mRNA codon to which it binds. The first mRNA codon is typically AUG. Attached to the end of the tRNA is the corresponding amino acid. Methionine corresponds to the AUG codon. The large subunit of the ribosome now binds to create the peptidyl, or P site, and the aminoacyl, or A site. The first tRNA occupies the P site. The second tRNA enters the A site and is complementary to the second mRNA codon. The methionine is then transferred to the A site amino acid, the first tRNA exits, the ribosome moves along the mRNA, and the next tRNA enters. These are the basic steps of elongation. As elongation continues, the growing peptide is continually transferred to the A site tRNA, the ribosome moves along the mRNA, and new tRNAs enter. When a stop codon is encountered in the A site, a release factor enters the A site and translation is terminated. When termination is reached, the ribosome dissociates and the newly formed protein is released.
Okay, any questions about that video? So let's talk about translation. Translation involves the ribosome. There's actually two parts to the ribosome, and they get together on the messenger RNA to make the complete ribosome. It involves obviously the messenger RNA, and it's said that the ribosome reads the messenger RNA. That's not quite correct. The ribosome comes together on the messenger RNA and then allows the tRNA to enter the ribosome, and the tRNA is what really reads the messenger RNA. And the tRNA brings the correct amino acid, and how that is happening is the anticodon binds to the codon. This actually is a hydrogen bond between UAC, which is the anticodon, to AUG, meaning A will hydrogen bond to U, U will hydrogen bond to A, G will hydrogen bond to C. And the ribosome stabilizes this link so that this anticodon, because there's only what, 2, 4, 7 possible hydrogen bonds that can even form. But the ribosome stabilizes it so the tRNA can come in and it will bind to the AUG. And then the tRNA will bring the correct amino acid. So this anticodon always codes for the amino acid methionine. And the tRNA will read the codons of the messenger RNA, bringing in the correct amino acid. And these are the components needed for protein translation. So the ribosome gets together on the messenger RNA, and it just moves down the messenger RNA, not doing anything until it reaches the start codon. Once it reaches the start codon, the first tRNA can come in, and it will always be the tRNA containing the amino acid methionine, because the tRNA will be the anticodon UAC, is the only anticodon that will bind to the start codon AUG. Any question about any of that? And that tRNA comes in this position. This is sort of like a hole in the ribosome, allowing the tRNA to come in. The second tRNA will come in the second hole in the ribosome and then bind to the second codon. And that anticodon will bring in the correct amino acid, which will be the amino acid for the second codon of the messenger RNA. Any question about any of that? Now, what's a little confusing is that the no, this isn't the confusing part yet. So, once this is bound, and that's bound, this first amino acid will be removed from the tRNA and then covalently linked to the second amino acid on the tRNA, meaning the second tRNA. What's removing this link between the tRNA and the methionine is actually, and then binding it to the second amino acid here, is actually the RNA enzyme on the ribosome that I mentioned is the only RNA enzyme we will talk about in this class. So what removes this methionine and then binds it to this amino acid is actually an RNA enzyme. And your textbook doesn't discuss it, but I do. It's the only RNA enzyme we'll discuss in the class. All right, now let's get to the confusing part, and that is once the amino acid is removed from the first tRNA, that tRNA will fall out of the ribosome, and then the ribosome will move down one codon, And what's a little confusing is that this tRNA then moves from the second position in the ribosome to the first position in the ribosome. Just trust me, it happens. I'm not entirely certain of the chemistry behind that equation, but it moves from this position in the ribosome to that position. That allows the next amino acid to come into the second position in the ribosome, and it will bring the correct amino acid that will correspond with the codon in the messenger RNA. So once again, the tRNA is reading the messenger RNA. Once again, the RNA enzyme in the ribosome will remove this amino acid from this tRNA and then link it to the newly added amino acid in the tRNA. Any questions about any of that? This tRNA then doesn't have an amino acid, or amino acids in this case, linked to it. And so this tRNA will fall out of position, but allow the messenger RNA to move down one codon. This will now move from the second position to the first position, allowing the next amino acid to come in the second position. And this creates a growing polypeptide chain as the amino acids keep being added to the added amino acid, meaning the latest newly added amino acid on the tRNA. And you can note here that AUG in this position is not coding for starting the protein. It's actually coding for adding the amino acid methionine. Anyways, the ribosome continues moving down the messenger RNA, moving in codon, one codon at a time, allowing the tRNA to come in, bringing in the correct amino acid, which will then be added to the growing polypeptide chain. And this will happen until the ribosome reaches the stop codon. The stop codon will code for the protein translation to stop. The ribosome will then dissociate and fall off the messenger RNA, and that will release the growing polypeptide, which is now, since the last amino acid has been added, it is now called the protein. You can call it a growing protein before the last amino acid is added, but after the last amino acid is added, it's now called the protein. Any questions about any of that? And there you see the ribosome falling apart into two pieces. The messenger RNA is now released. The tRNA is also released, and the protein is released. Any questions about any of that? If not, let's talk about a little bit of complication, and that is here we can see that there is more than one messenger RNA being transcribed on this one gene. So here's the gene here, something like there. And you can see 1, 2, 3, 4, 5, 6, 7, 8 messenger RNAs being made on this one gene at close to the same time. Obviously, this one started first, and that's why it's longer. And then that's the next, well, that one you can't see, but that's the next longest. These are shorter, 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 and there's the newest one. It's the shortest. Any question about that? So this complication can happen in both eukaryotes and prokaryotes, and that is you can have more than one messenger RNA being transcribed on one gene. In prokaryotes, we have another complication, and that is transcription is occurring in the cytoplasm, And you see these nodules here? Those are actually ribosomes being bound to the messenger RNA. So this short messenger RNA, meaning the messenger RNA isn't even finished. There's 2 ribosomes already bound to it, and they're going to start making the growing protein. And this one here has many ribosomes bound to it, and that one probably would be the first, so it would be, or is that one the first? Let me think about this. It's going to start here and then move that way. So this was the first one that was added, and so it'll be making the proteins. My point is that in prokaryotes, you can have protein translation occurring before transcription is finished. We are transcribing the gene. This might be the end of the gene, but these are definitely not the end of the gene. And protein translation has even begun on this messenger RNA, which this is only a part of the small part of the messenger RNA. And that's because transcription and translation can happen at the same time, because in prokaryotes, this is happening in the cytoplasm. In a eukaryote, transcription happens in the nucleus, translation happens in the cytoplasm, and so the two cannot happen at the same time. Transcription has to finish before protein translation can occur in a eukaryote cell. Any question about any of that? Anyways, there's the complications going on. That is, more than one messenger RNA can be made from one gene, and then you can have multiple proteins being made, at least in a bacteria or a prokaryote cell. You can have multiple proteins being made on the messenger RNA before the messenger RNA is even finished transcribing. In eukaryotes, we have another complication. This doesn't happen in bacteria, but in eukaryotes, we have exon regions of DNA that code for the protein, and we have regions in the gene called introns that do not code for the protein. When the first RNA transcript is made, it's going to have both exons and introns. The introns will prevent this initial RNA transcript from leaving the nuclear membrane, meaning the pore here has a check, and if it finds introns in the messenger RNA, it will not allow that messenger RNA to leave the nucleus. The introns have to be spliced out. Splice just means cut it out and then put the exons back together. That's called splicing together. And so cut out the introns and then splice the exons back together. And then the messenger RNA transcript minus the introns can leave the nucleus. So the pore will notice this transcript has no introns and allow it to leave the nucleus. Once it leaves the nucleus, it's in the cytoplasm, and the ribosomes can bind to the messenger RNA and then engage in protein translation. Any question about this complication? All right, so we talked about the structure and function of genetic material, the flow of genetic information. We talked about DNA replication, RNA transcription, and we're just finished protein translation. Let's move on and talk a little bit about the regulation of bacterial gene expression. I'm going to do that by talking about operons. On Thursday, we'll talk about mutation, genetic transfer and recombination, and genes and evolution. Do we have time for that? Yeah, we do have some time on Thursday. We're talking about chapter 8. I'm going to warn you that I'm going to have to leave about 6:10, so I'm going to close the class a little bit early. So just be expecting we're going to end the class a little early. I've got something I have to do, and I don't have enough time to do it with between the lecture and the lab, so we're going to end class a little early today. But let's talk about regulation of bacterial gene expression. There are two main ways to regulate what is inside a cell. You can control gene expression, meaning if you don't want a product made in a cell, you can turn off the gene, and that will not allow the product to be made. And then another way you can control what's in a cell is by controlling the post-transcriptional control. Like, for example, if you didn't want to make the protein, you could degrade the messenger RNA, which codes for that protein. That's post-transcription. I mean, you made the messenger RNA, now you don't want the product of that messenger RNA, and so you can degrade the messenger RNA. There's other ways you can do post-transcriptional control. You're not going to be tested on it. We're not going to talk about it. But one way you can do it is you can inactivate the protein. And then so the protein won't be active in the cell. We're going to talk about controlling gene expression. There are some genes which are expressed at a fixed rate. So these are called constitutive genes or constitutive enzymes. So this enzyme is made at a fixed rate, and it's always made in the cell. There are other enzymes that are only expressed as they are needed. And these are controlled by repressible genes or inducible genes. So we have a repressible enzyme or an inducible enzyme. And these enzymes are only made when they are needed. You can regulate a gene by pre-transcriptional control, by an inducible operon, and we'll talk about that. You can also regulate the gene by a repressible operon. We're not going to talk about that. It's similar to an inducible operon, just a little different. We're not going to cover it. You're not going to be tested on it. And then there's an epigenetic way to control the gene. And we're not going to talk about it. And fortunately, you're not going to be tested on it because geneticists don't fully understand epigenetic control mechanisms. And they've been working on this since at least the 1950s. And they don't have a real good understanding or a real good idea of what's going on. Okay, fortunately, we're not going to talk about it. So genes are not always turned on all the time. If the cell doesn't need that gene, it can turn that gene off. How the cell does that is using the operon. An operon model of gene expression is a gene, let me blow this up. A gene encodes a structural gene, and normally when we talk about a gene, we're talking about the structural gene. Like when we're talking about the gene for eye color, for blue, that is a structural gene. When we're talking about the gene encoding the enzyme lactase, that is a structural gene. In fact, in the lac operon, the Z gene, the Z structural gene, codes for the enzyme lactase. In bacteria, you can have in an operon more than one structural gene. But if there are more than one structural gene in the operon, all of these genes are related to each other, meaning in the lac operon, this is this operon here, all of these structural genes are involved in the metabolism of the sugar lactose. The Z gene encodes for the lactase enzyme, which will degrade the sugar lactose into beta-galactosidase. galactose and glucose. Oops, sorry, didn't mean to do that. The Y gene is the protein transporter, transport protein in the membrane that allows the sugar lactose to gain entry into the cell. So the Y gene allows the sugar lactose to move through the cell membrane. Come on, mouse. And then the A gene is involved in the metabolism of beta galactose. It's involved in converting beta galactose to the sugar glucose. So all three of these genes are involved in metabolism of the sugar lactose, and that's the way an operon is. All of the structural genes, if there's more than one, will be involved in the same process. We'll word it that way, meaning all of the genes in the lac operon are involved in the metabolism of the sugar lactose. All right, so that's the structural genes. In the operon, we also have other DNA regions which control the structural gene, control the expression of the structural gene. One of them we've already talked about. That's the promoter. The promoter codes for the binding of DNA polymerase to the DNA. And when the DNA polymerase binds to the promoter, it'll then move downstream, and it will eventually transcribe the structural genes, especially the Z gene, the lactase enzyme, or encode for the lactase enzyme, lactase. The other controlling region is the operator, and it is involved in controlling whether this operon is turned on or turned off. And then there's also a small region at the end of the gene. It'll be the terminator. Eukaryote operons are similar to prokaryote operons, except they only have one structural gene in each operon. And so, I gotta leave here. So in eukaryotes, we would have three structural genes for the lac operon, and one structural gene would encode the enzyme lactase. The other will be the gene Y, and then the third will be gene A. There is one other thing where eukaryote cells differ from prokaryote cells, and then that is they have more regulation than a prokaryote cell has. We're only going to talk about the regulation of prokaryotes, but they do have more regulation. All right, any questions about anything so far? We'll continue this lesson on Thursday. All right, if there's no questions, I'm going to say goodbye, and I will see you in the lesson 6:30.